So, Professor Issam Awad is the John Harper Seeley Professor of Neurological Sciences, and he's Director of Neurovascular Surgery at the University of Chicago. He is a world leader in neurosurgery. He's authored over 400 publications, that's a lot, and led pioneering research into cerebral cavernoma malformations. And he's going to be talking to you today about the AT-CASH EPOC trial with statins and cavernoma. And as Professor Rustam Al-Shahi Salman talked about, these trials are so important because we really want to improve clinical care for people with cavernoma. And as I said, just as we finished that session, for those who weren't there, you can make all the difference by being research trial participants and really help to improve outcomes for the next generation of people with cavernomas.
Thank you. First of all, it is really a pleasure and an honor to be with you. I recall traveling nearly 20 years ago to London for one of the first meetings that the Angioma Alliance UK had, which we held in person, and I remember it quite well. And it's been a long time, and there have been a lot of discoveries, but also a lot of activities and engagement by patients and communities across the Atlantic. And I am a fan of the Angioma Alliance UK, and I have a number of patients with whom I interact in various ways. And of course, many colleagues, particularly Professor Salman, with whom we collaborate a great deal on many aspects of trial planning and on the next phases of therapies for this disease. What I'm going to speak about today is a particular trial that is a first-in-disease trial, so a lot of lessons were learned and a lot of new concepts were introduced in conjunction with this trial. And I'll walk you through some of these that took approximately 10 years to plan, plus another 5 years to execute. So the trial was completed with the last patient seen in July 2024, and the results were published in March 2025. So you will hear now in detail the background of this trial and the results. So the study was sponsored by the United States National Institutes of Health. One additional source of support was a private donor who helped us design a smartphone application, which came in quite handy during the COVID pandemic, and you will see the incredible results that it helped us accomplish in terms of compliance monitoring and enhancing compliance with the drug during 2 years of the trial that were running, including during the pandemic. I work as a consultant to drug companies that are trying to bring drugs to patients. These did not have any involvement in this trial. And similarly, I render occasional legal consultations on CCM disease. The trial, as I mentioned earlier, was first presented at the International Stroke Conference in February 2025. And this is the trial team from University of Chicago and Johns Hopkins that are celebrating that day. And it appeared in the Lancet Neurology in March 2025. So the whole concept of symptomatic hemorrhage in cavernous malformations is important to start understanding what was behind the trial. So a symptomatic hemorrhage is a cardinal event, and it involves new blood emergence attributable to this bleeding. So it is this particular patient had the new onset seizures as well as headaches with unmistakable new bleeding in one lesion while all the other lesions in the brain were unaffected. This is a familial case. So this would be the symptomatic hemorrhage, and this would be the CASH lesion or cavernous angioma with symptomatic hemorrhage. What's unique about this is that once a lesion suffers such a symptomatic hemorrhage, we know now from many studies, but the key study that I like to quote is the population study published in 2012 by Professor Salman. And it shows that once a lesion has suffered a symptomatic hemorrhage, for up to the next 5 years, the risk of bleeding is quite high, is more than 6% per year, while a lesion that hadn't suffered, like all these other lesions, will have less than a fraction of a percent per year bleed rate. So it's more than tenfold increase in the risk of hemorrhage. So this is a ticking lesion that changed character compared to all the other lesions in the same brain of the same patient. So, and therefore, this is a particular target for therapy to prevent rebleeding because of its high rebleeding rate. And we currently have no proven treatment. We, of course, attempt surgery to resect such a lesion so it doesn't bleed. But if the lesion were in a more eloquent portion of the brain or in the brainstem where surgery might have a much higher risk, then surgery is really associated with risks and costs. And everybody would love to have a pill rather than or some drug or other treatment rather than surgery to prevent the rebleeding risk. So the biologic rationale for the drug that we tested has to do with Rho kinase, which is a molecular signaling aberration in endothelial cell that results from the loss of the CCM gene. So once we have a lesion, a cavernous malformation lesion, it typically has a loss of function of one of these genes within the endothelium of the lesion. And once there is a loss of function in one of these genes, there is an increase in Rho kinase activity, and this renders the endothelium permeable and incapable of forming tubes, which we want the capillaries to be tube formation. When they have a Rho kinase increase, they are incapable of forming tube, and instead they form bubbles. So it becomes a small cavern, and actually Rho kinase increase also promotes further growth and further bleeding in the lesion. So if we inhibit Rho kinase activity with a very specific drug that is not available to humans for chronic use, it's called Fasudil. It's a very effective blocker of ROCK. You immediately have about an 80% reduction of lesion burden over the next several months in the mouse models of cavernous malformation, but also interestingly, a decrease in bleeding in the lesions themselves. So decrease in size and number of lesions as well as decreased bleeding. What is interesting is that statins, which are commonly used for cholesterol lowering, at high doses, they also have Rho kinase inhibition properties, which is thought, by the way, to stabilize the atheromatous plaque in the coronaries and in the carotid arteries and elsewhere. So the statins do a lot of good for atheromatous plaques, partly by inhibiting Rho kinase, but you have to have a high dose statin. So simvastatin, which is a lower dose drug, accomplishes mild effect in bleeding and no effect on lesion burden, while atorvastatin at a dose equivalent in mice to that used in humans for cholesterol lowering, which is 80 milligram per day in an adult human being is equivalent in mouse to 80 milligram per kilogram per day. And that dose accomplishes the beneficial effect in the mice on lesion size, as well as non-heme iron deposition, which is the type of iron that is deposed when you have new bleeding. However, the use of statin has been controversial in cavernous malformations. Some people have raised the concern, might it cause worsened bleeding? And in fact, some people quoted biological reasons that it could possibly increase bleeding. There are studies in some stroke, hemorrhagic stroke trials where the use of statin may have increased bleeding risk. Therefore, there is a concern about just removing the dam and letting everybody use statins. So we have first to know that it is safe, and we don't know that it actually works in humans as it does in the mice. Now, the problem with testing a drug, any drug, and you heard that from Professor Salman before me, any drug on cavernous malformation with the aim of altering symptomatic hemorrhage rate has to do with selecting lesions that have a high risk of hemorrhage. And even then, you have to have a strong effect on the bleeding rate in order to have a reasonable size trial. So as you would see, if the bleeding rate is somewhere between 6% and 15%, you need hundreds of patients for more than a year to show a 25% effect, and even hundreds of patients for more than a year to show a 50% effect. And 50% effect of a drug is quite magical. I don't think we would see that. So it in a rare disease like symptomatic hemorrhage with cavernous malformations, it is almost impossible to conduct trials powered to detect an effect on symptomatic hemorrhage rate, even if we knew how to pick lesions that have a 30% hemorrhage. And to date, we don't have any trial readiness study that shows a subgroup that has this high a risk. But even if we had this high a risk, it would take hundreds of patients to have an effect. So we need a surrogate biomarker of bleeding to test a proof of effect before we put hundreds of patients on a drug for many years. So that was a problem, and we had to overcome this problem with several years of research studying what happens on the MRI of the brain to a CCM lesion when it bleeds. And as it turns out, when a lesion bleeds, you see the new blood, of course, but also at the same time, if you measure permeability in the lesion, the permeability increases. And if you measure the amount of iron, and we have a method that is called quantitative susceptibility mapping, you are going to hear a lot about it, that actually is a MRI scan without gadolinium that allows you to measure the amount of iron in every pixel, in every dot of the lesion. So you could do an average or a mean QSM for the whole lesion. And that mean QSM increases every time you have a symptomatic hemorrhage. And it also, so every time you have a symptomatic hemorrhage, it increases. Permeability increases, but 80% of the time. A symptomatic hemorrhage increases QSM 100% of the time. And even when there is asymptomatic bleeding, QSM increases. But also QSM happens to increase silently without symptoms in many lesions. Therefore, as a surrogate measure of bleeding, the amount of iron in the lesion can be targeted by a drug. And if we really look at the effect size on QSM change in the lesion, now we can have situations where the number of patients per group is very doable, 25 patients, even if you allowed attrition rate or no follow-up date or non-compliance rate, you end up having 40 patients per group. That's 80 patients in the trial. In this case, they are followed for 2 years, and they are assumed to have no dependence between them in year 1 and 2. If there was some favorable dependence, you could even have the effect detected in fewer patients. So this is a 20% effect size. So this is a very favorable thing. So you could do a trial, and a mild change in iron in the lesion would be detectable with a few patients. So this allowed us to reach out to the United States Food and Drug Administration, and they have approved, in fact, QSM change as a surrogate biomarker of a drug effect for future drugs being developed that the FDA might provide an IND for. So with these things in hand, we received funding from a National Institute of Health to conduct this trial. And the trial was called AT-CASH EPOC, so atorvastatin treatment in cavernous angiomas with symptomatic hemorrhage, exploratory proof of concept trial. It is exploratory proof of concept because it's the first time we're using a biomarker, and we designed the trial accordingly, with the primary hypothesis is that the intervention impacts 20% difference in the annual QSM percent change with 2 annual epochs of measurements. So the patient would take the drug for 2 years. They would have an MRI at the beginning and end of each year. So 3 MRI scans during the trial. And the trial is two-tailed. For those of you who don't know statistics, it is designed very rigorously to test whether there is increased bleeding or decreased bleeding. So it's not designed with a bias toward benefit or harm. So it allowed us to test either way, and with a very strong power for a phase 1/2 trial, a power of 0.9 and with an alpha of 0.05. So those are very strong thresholds. We decided to go for this rigor because we didn't want a result on a biomarker that may be positive, but with weak statistical power or weak significance. So if we have a result that's significant, it would be believable. The trial required travel to Chicago because at the time of the initiation and funding of the trial, the QSM technique was not validated at multiple sites. So we did not want the QSM technique to jeopardize the quality of the data. So we provided travel funds for each subject. And in fact, about 70% of the patients traveled from outside the Chicago area, and they traveled again at 1 year and 2 years for the trial. And of course, we monitored their compliance throughout the year with the application remotely, and also worked with their local physicians if there was a question of rebleed, so that they would receive an MRI, and then we would act on the rebleed. We were particularly proud that we were able to enroll a large number and retain with higher enrollment rates and higher retention rates of African Americans and Hispanics and women in this trial, and we published that separately. The trial, as we said, aimed to enroll 80 subjects randomized equally to atorvastatin and placebo. This is the 40 inflated sample size. We would hope that we would have actual results on about 25 in each group in order to meet our statistical significance or statistical power, and we accomplished that. So basically, we enrolled 80 after pre-screening 300 and some subjects. And once patients were assigned to atorvastatin or placebo, some of them, of course, dropped off for the expected reasons, and some withdrew from the study. We are actually proud that we kept the withdrawal rate at under the 30% number that we had projected. Despite the pandemic, we ended up having 33 patients in the atorvastatin group and 31 patients in the placebo group that contributed more than one or more paired QSM assessment. So we would call those the modified intention to treat cohort, and based on those, we analyzed the QSM change in treatment versus drug. Of course, symptomatic hemorrhage rates were also monitored, and they were monitored in the whole sample size in all the patients. So we counted every bleed, even if a patient had a bleed and did not come back for a paired QSM assessment. So we counted all bleeds regardless of whether they had paired QSM assessment. We are particularly proud of this compliance rate, I think, for any drug trial going over 2 years, where patients read a lot about possible side effects on Wikipedia, and are my leg pain due to my statin? And it turned out that I will show you the data. Many of the patients had the same complaints, whether they were on placebo or atorvastatin, but we're proud of them because they stuck with the drug, and we did not have any serious adverse events that required cessation of the drug. The 4 patients that were non-compliant, one of them had rebled and stopped the drug, and 3 others just were non-compliant and withdrew from the study. So they were not part of the modified intention to treat. So more than 90% compliance rate, counting every drug dose every day for 2 years. The baseline features of this trial were extremely well balanced, and in almost every aspect, including the use of medications like blood thinners, vitamin D, their lipid levels. None of them were on any statin because that was an exclusion from the trial. So they were started on statin for the trial. The only imbalance, more patients that were disabled at entry into the trial in the atorvastatin group, who ended up randomized to the atorvastatin group, and fewer of those requiring some assistance in daily living in the placebo group. So this is unfavorable to the drug, and we correct for it and factor it later on. The actual quality of life assessment by the patients themselves were quite well balanced between the 2 groups. We were very careful to balance for the use of vitamin D and for sex hormones, which have been suggested as potentially altering bleeding rate. And we also did not want an imbalance in the use of propranolol, which, as you know, was being tested for possible effect on CCM, and we did not have any imbalance there. So any effect that you see or don't see between the 2 groups is really due to the drug itself. The primary outcome was the point effect was very slightly in favor of atorvastatin, and the effect size was merely 10%, which is half the effect size postulated to achieve statistical significant. So if you had had a 20% effect size, we had the power for this to become significant, but there was no, we did not reach that difference. So the effect was very weak. And of course, this is not statistically significant, so it may not have been real. And when we imputed for missing QSM results, so there were patients that bled and did not have a QSM, so we imputed the results in a very sophisticated technique. This did not change the outcome. When we looked at effect on QSM in every year, and when we looked at it at absolute change versus percent change, also there was no benefit. The permeability actually was not helpful at all. The point estimate favored placebo, but in fact, the variance rates were so great that there was really no way that an effect on permeability would have been detected. This was a secondary outcome anyway, and it had nothing to do with the primary outcome. We did learn that many of the patients who had the QSM change also had the permeability change, but the permeability change had such great variance between patients that the effects were all over the place. This is very interesting. This is the stuff that everybody is familiar for, which is counting symptomatic hemorrhage in all the patients enrolled, 40 in each group. And the symptomatic hemorrhage, if we had followed the group for only 1 year, it looked like it was favoring a placebo in a dramatic way. So there were favoring atorvastatin in a dramatic way. So there was only 1 bleed on atorvastatin and 4 on placebo. It was not statistically significant, but using the 80% confidence interval, it may have tipped someone to think this may be a good outcome. The only problem with this is that the biomarker change, the QSM change during that time, did not support that big difference. And because patients were followed for 2 years, and some of them checked in at the 1-year follow-up, and we found that they had bleeds during this year, so the bleed was counted here, you started having a greater number of bleeds in the 2nd year in the atorvastatin group. So at the end, in the 2nd year, the effect was the other way, more bleeds in the atorvastatin than the placebo. So we would very strongly caution against this 80% confidence interval as a decision maker. It is a signal that one should verify, but it is not one that, in our opinion, justifies making big decisions on stopping trials or anything like that, because you would end up by 1 additional bleed here in the study, it would wipe that effect out. So it's a stochastic, statistical. There are so few bleeds that this was not sufficient. And this tells us that a study powered to detect an effect on symptomatic hemorrhage, it really requires a large number of patients in order to be reliable. So pre-specified measures of harm, also changes in QSM, symptomatic hemorrhage rates, or symptomatic or serious adverse events requiring drug discontinuation. Those really did not, were not any different or any greater in the atorvastatin. So there was really no concern about harm whatsoever. However, the symptoms reported by the patients were more frequent in the atorvastatin group. So the number of patients reporting symptoms is only slightly higher. So 21 in the placebo group reported side effects, 27 in the atorvastatin reported side effects. But look, these patients reported more side effects. So I'm tired and have leg pain, or various other or GI symptoms or diarrhea. And these were all mild symptoms, not enough to stop the drug. but they were significantly different when you factor both the number of patients and the number of events reported by the patients. Mostly they were myalgias, diarrhea, and tiredness. When we looked at the ROCK inhibition in the blood, we were actually quite surprised that in the blood the effect in the blood itself, cholesterol was lowered by atorvastatin, which is here at the baseline they were the same, but at year 1 and year 2 the patients had lower cholesterol, which is really what you would want to see in a drug that lowers cholesterol. However, Rho kinase was not affected by the drug the way we would have seen if the patients had atherosclerosis. And the difference is that our placebo patients had relatively low Rho kinase activity. In all the trials of heart disease where atorvastatin lowers ROCK, they started with very high ROCK levels in the placebo, and atorvastatin shut it down. But when you start with healthy patients who do not have high ROCK activity, the effect on ROCK in the blood was not significant. When we measured brain white matter permeability, which is a surrogate effect of Rho kinase inhibition, it was decreased by the drug, but the decrease was not statistically significant. But you could see this is quite a robust effect. If we had more patients, it would have been significant. So as a biomarker signal, it was working, but it was weak. Effects in pre-specified subgroups were interesting. Of course, you start now splitting an already small group of patients, but we were surprised to see that in brainstem lesion location, the effect favors atorvastatin more greatly, and in the non-brainstem, it actually does not favor atorvastatin. So that if the whole trial was performed in brainstem patients only, it would have been significant. The effect would have been more than 20%, and it would have been significant. So there are many reasons why this could be. Possibly that many of the serious bleeds outside the brainstem did not enroll because they ended up having surgery. So we ended up enrolling milder bleeds outside the brainstem, and the brainstem bleeds were more serious. The slight bias favoring males and favoring sporadic lesions actually can be accounted for by the brainstem location biases. So the brainstem location effect is the only one that is independent of sex and genotype. So in conclusion, we have no concern about the safety of atorvastatin in CCM patients after recent symptomatic hemorrhage. There was no increased bleeding by any measure. This is, we believe, an important finding clinically for patients who must be on or should be on cholesterol lowering drugs. For cardiac indications, they should not fear that atorvastatin at high doses harms their CCM lesions. We have no evidence of harm whatsoever, and this is helpful. But also, there was no benefit in decreasing bleeding risks as postulated, and we cannot justify atorvastatin use for this indication. As I indicated to you, the point estimates of a weak, non-significant benefit are consistent with weak ROCK biomarker effect. So they would justify the question, would a stronger, more specific ROCK inhibitor like those being developed now by the pharmaceutical companies targeting ROCK2, which is more brain specific rock and without the pleiotropic effects of lowering cholesterol and other effects of statin. So these stronger ROCK inhibitor, would they achieve the desired benefit? That is the question that is still out there. The direction of the point estimate in the subgroups suggests the need for stratifying for those subgroups in future trial, particularly brainstem lesions and maybe a futile in the non-brainstem lesions, and the trial would continue on only brainstem lesions. So my last slide is really both an acknowledgement and a celebration. This was a partnership with my trialist partner, Daniel Hanley at the Johns Hopkins Medical Institute. He handled all the data management aspect for this trial, and it was the data coordinating center. We had the contracting system for the data entry remotely. The biostatistics were done by a wonderful senior statistician at Johns Hopkins who had worked with me previously on other brain bleeding trials, Rick Thompson, trial manager and trial nurse working side by side with us were extremely helpful. The trial nurse was open for the 4 years of enrollment of patients and their follow-up in the trial. Kelly Fleming from the Mayo Clinic was an independent safety monitor. Other safety consultants were there, but as we said, we did not need their help because there were no safety issues that arose. The co-investigators helped a great deal in fine-tuning the biomarker acquisition, the quality of the biomarker work, as well as the trial analyses. Imaging and bioengineering was, of course, a big aspect. And as a result of this trial, now we can deploy QSM effectively at multiple sites, and we have multi-site validation studies that have taken place. I also failed to mention that in this trial, we did a mid-trial adaptation of sample size, and the sample size was right on. So the failure to show an effect was not because we had too few patients. It was because the effect was weak. And also we did a futility analysis and safety analysis throughout the trial. Statin clinical care was overseen by a senior cardiologist, and the ROCK assays were performed by the person who invented the plasma ROCK, the blood ROCK assays on peripheral white cells. Fellows and students were involved in this, including a fellow who was the primary author on the paper with me, and the referral network through the Alliance to Cure and the CASH Consortium trial readiness group, as well as StrokeNet supported by the National Institutes of Health. So I think this trial basically moved us from a biologic plausibility to biomarker science to novel statistics design and innovation and infrastructure that will be useful in future studies, and is now free to the world because it was sponsored by the National Institutes of Health. So this is all public discovery information for drug companies and other parties in Europe and elsewhere to use all these tools that were developed and fine-tuned in this trial. So I want to thank you and stop sharing here, and I hope we have a few moments for questions. I don't know, I must have overstayed my welcome.
No, you couldn't at all, Professor Awad. That was absolutely fascinating, and we do have time for questions. And I just want to say how important this is, because for this trial, there are a lot of clinicians very worried about prescribing statins and patients worried. So an important outcome of this trial is that people can have confidence taking them. And as you say, there's so much else we can glean from that. Personally, I was quite interested about the brainstem cavernomas and the people in the trial. I'd be interested to see what happens next. So for everybody who's joined us, as I said in the chat, Professor Awad is globally recognised for his expertise in cavernoma. So now is the time to ask questions, and I've already had some come in. So if you don't mind, I'm just going to move to those. So the first question is, and just to reiterate to everyone, Mr. Awad can't give clinical advice specific to you. However, he can talk in general terms. So this question says, and it's from Ian, who is our founder. He said, "Thank you so much for your presentation. Fascinating." A question.
Hello, Ian. How are you?
He is good and still very involved. And he said, "Could you please say whether CT scans reveal only fresh blood from a cavernoma, whereas MRIs show old blood staining ad infinitum?"
Well, it is true that CAT scans would show new bleeding in cavernomas. However, there is some subtle bleeding in cavernomas that is very hard to glean on CAT scans, because the CAT scans also have calcium from the lesion that blocks our ability to see new blood. MRIs are more sensitive at detecting new bleeding as well as old bleeding. So if you have new symptoms, MRI will show new blood. MRI does not only show old blood. But as the blood turns from new to old, the MRI will also show that. And mostly through this quantitative mapping of iron, not only do we see more blood in the hemosiderin ring, but even if the hemosiderin ring does not change, we can measure how dark it is using the QSM technique.
Fantastic. So we've just had someone say in the chat here, "Thank you so much for doing this research. It helped me make a decision on whether to take statins or not." So the next question is from somebody who's getting a lot of urinary tract infections. And they say that they think they saw you'd mentioned somewhere that these could be dangerous for cavernomas. And they're asking what can they do to stop them, and how dangerous are UTIs for people with cavernoma?
Yes, this the basis for this observation and then recommendations is a fundamental biologic discovery, in that the gram-negative bacteria in any form will enhance bleeding from cavernous angiomas in mice, for sure. And this was such a fundamental discovery that was published in the journal Nature. By Tang et al. in Nature, and then subsequently we show.
Apologies, everyone, for the sound issue. So, Professor Awad, can you still hear us? We're just having a couple of connectivity issues.
Propensity in cavernous angiomas in human communications. So these.
Oh, so sorry. We had a little bit of connection issues there, but what I did hear was you talked about gram-negative bacteria enhancing bleeding risk, and this had been published in the Nature journal, which is a prestigious journal, and then we just lost you at that bit.
Great. So that was by Tang et al. in Nature. And then subsequently, there was a paper also showing greater gram-negative bacteria in the microbiota of patients with cavernomas and with active bleeding cavernoma. And this was by Polster et al. In Nature Communications. So also very rigorous work. So there is a lot of fundamental biology. Now, the number of patients with urinary tract infections causing a bleed is merely anecdotal. We see it in our clinic all the time. And the way we see it is every patient that comes to my clinic with a bleed, we go over potential triggers. What might you have had? What might you not have had? A urinary tract infection, as well as stimulant drugs or sex hormone drugs or whatever emerge in many patients. And these are anecdotal associations. They are not proven in prospective studies, but they are very congruent with the biology of the disease. So in the mice, if you have a gram-negative infection in the belly or the urine of the mouse, it will trigger cavernoma bleed. So it's a very, very fundamental biologic phenomenon. I cannot tell you that this happens in every person with a UTI, but I have seen in my practice many patients where there was no other explanation for the new bleeding but a significant urinary tract infection preceding the hemorrhage. So as Professor Salman said, you should treat a urinary tract infection anyway. So if you have a cavernoma, you should treat it even with greater vigor.
Well, I've learned something new today because I didn't know about
I
UTI.
hope that helped.
So, Professor Awad, just to flag, we are having slight connection issues, but we can still hear enough.
Here you go.
And we've got a couple more questions which I'd really like to ask. So we'll see if we can quickly ask those. So somebody has said, we're a family with cavernomas, but the genetics can't establish the gene. Should we be monitored more closely, as at 61 I have too many cavernomas to count, and I have not been offered any follow-up checks with MRI?
Well, you know, in the United States, we do recommend follow-up MRI to assess lesion burden with the understanding of to look for triggers, because
so I'm so sorry, everybody. Unfortunately, the sound quality has got very poor.
Things
Professor Awad, if you can hear me, unfortunately, we can't make out too well what you're saying.
And people have things lives that happen, or they get on sex hormone drugs.
Professor Awad? So I'm so sorry, everybody. Professor Awad was ever so kind and joined us from his holiday. But it does mean then the internet connection can be a little bit intermittent, and we have lost him there. But I think we have all learned so much from that presentation, and I'm so glad we got an opportunity to hear from him for certainly a few questions.